Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 6.3, transcription and RNA processing. Last lesson we talked about DNA and DNA replication, and today we're going to start adding RNA into the processes that we'll be discussing this unit. And so we're talking about the mechanisms by which genetic information flows from DNA to RNA to protein. And today we'll be focusing on the specific stage known as transcription, in which DNA becomes RNA. And so we'll be talking about the different kinds of RNA, as well as the process by which transcription occurs, and suggest a scale of visual representations to be explain relationships between characteristics of biological models in both theoretical and applied contexts. So the majority of this unit focuses around the idea of gene expression, which is the process by which DNA directs the synthesis of proteins. So what proteins are made depends on what your DNA is. And each gene codes for one polypeptide. So each gene is going to code for one protein. And the genetic code is nearly universal, which is, again, evidence for common ancestry. And so we're going to be talking about the central dogma, which is the idea that DNA exists and it becomes replicated to preserve this hereditary information. Through transcription, it becomes RNA, and then through translation, this RNA will code for the production of a protein. And so today we'll be talking about transcription, which is the synthesis of RNA using the DNA as a template within the nucleus of eukaryotic cells. And our goal is to generate mRNA, messenger RNA, which carries genetic messages of DNA to the protein-making machine in the cell. So carrying this genetic information to ribosomes, because the DNA is not leaving the nucleus. Messenger RNA is the one that is carrying this genetic information over to the ribosomes so that translation can occur and a protein can be produced. So, in transcription, one strand of the DNA is transcribed. This is the template strand. That's what the mRNA is going to be based off of. So if we look at this picture right here, we see our template strand of having a thymine, an adenine, and a cytosine molecule. So RNA is going to be basing off of that, and it's going to be creating base pairs to those nucleotides in the DNA strand. And the other DNA strand is known as the coding strand. It's going to have the same sequence as the mRNA because both of these were base paired to the same template strand. And the only difference is that RNA is going to contain the uracil molecule while DNA is going to have the thymine molecule. And so the enzyme, again, that we're talking about here is RNA polymerase. This is the enzyme that first is going to separate the two DNA strands and then connect free RNA nucleotides, meaning just free nucleotides, but of course focusing on having uracil instead of thymine, and then as they base pair, adding them to the 3' end. And so DNA is going to be read from this leading from 3' to 5' carbon so that RNA can be added 5' to 3'. And there's going to be a promoter attached to the DNA sequence that is going to then promote RNA polymerase to attach and start unwinding the DNA and adding these nucleotides. And then it's going to detach at the terminator. And transcription factors are going to assist in this process of promoting RNA polymerase to begin transcription. So transcription is all about just adding base pairs onto one strand of DNA in order to create a single strand of RNA. And so this process creates primary RNA that must be processed before it becomes mRNA. And this processing is only going to occur in eukaryotic cells. In prokaryotic cells, transcription and translation happen pretty much at the same time. And so the RNA is going to be processed by the addition of a 5' cap, which is a guanine nucleotide attached to the 5' end of mRNA, and a poly A tail, which is 50 to 250 adenine nucleotides attached to the 3' end. And so these are going to assist the mRNA in exporting from the nucleus through the nuclear pores, and they're going to protect the mRNA from degradation, as well as help the mRNA attach to the ribosome to begin translation. And the poly A tail is specifically helpful for protecting from degradation because in the case of nucleotides accidentally being taken off the mRNA, there's going to be 50 to 250 adenine nucleotides that are not coding for any sort of protein that can be removed from the mRNA before the coding sequence is affected. And depending on how long this poly A tail is, that's going to determine how long the mRNA should last. Another very important part of mRNA processing is RNA splicing, which are when specific sections are removed. These are the non-coding sections that are removed, known as introns. We discussed them in 6.1. And the remaining are exons, and they're bound together by a spliceosome. And so the cool thing is that an intron in one RNA can be an exon in the other. So even with the same with the same strand of DNA and portion of DNA that has been transcribed through RNA, it can still result in differing gene expression, differing protein synthesis, because what is considered the coding and non-coding regions could change in different situations, and this is known as alternative RNA splicing. And there's a special RNA in spliceosomes known as small RNAs, which are ribozymes. And these are enzymes that are made out of nucleic acids, not out of proteins, as we initially thought all enzymes were, which is pretty cool. So over here you can see the primary RNA transcript, and through RNA processing, the 5' cap is going to be added, the poly A tail is going to be added, and these introns are going to be removed, and then the rest of the exons will be bound together by a spliceosome. So there you go. Remember the central dogma, DNA to RNA to protein. And today we discussed transcription, talking about RNA polymerase, the different base pairs that are formed, and how mRNA comes about from a strand of DNA so that this mRNA can then be processed and exported out of the cell in order to create a protein. Next up, we'll talk about translation, which is reading this mRNA and turning it into a protein.